Oh, thank you, Eric, for telling me that I'm a great instructor. Thank you so much. When is the math class? The math class is tomorrow. We have the schedule on kidteachkid.org under the summer camp schedule part, and you can just see that. We have some great math instructors, including Rich, who is right here. Yeah. All right, so we're gonna be starting biology now. Yeah, it's great to see so many of you guys come. It's a great way to spend your summer. Thank you so much. All right, so for biology, today we're going to be talking about biochemistry and molecular biology. In the beginning of the class, you guys may be like, what the heck? Why are we talking about, when is class over? If you're just now coming in, the biology class is from 5 to 6. But if you just finished the public health class, it's basically done. But if you want to stay for biology, feel free to do that. All right, so biochemistry and molecular biology is what we're going to be talking about today. So in the beginning of this class, as I was saying, it's going to be a lot of chemistry since a lot of biology is centered on chemistry. We're going to be covering a lot about how chemistry integrates with biology. And then at the end of the class, or near the end, we're going to be discussing cells, like basic concepts of cells. I think someone, oh, thank you for sending my magazine feature. All right, anyway, so we're going to start now. Yeah. Oh, and I think one more thing I forgot to mention. There's also homework for public health if you're left. Sorry, but you don't have to do it, but it'll give you points. All right. So here's another little plug. If you did not go to my public health class, basically I published a book on epidemiology, and it's good for helping you understand the COVID-19 epidemic or pandemic. Sorry, and then it's there's a link. There's a link. I can send it into the chat right now. I believe it's also on the Kid Teach Kid website. Let me just send you guys the link real quick to buy the book. What the, wait, hold on. My computer is kind of lagging. But anyway, actually my computer is lagging, so I'm sorry. I'll just send it later. But basically, you can just go on lulu.com and search epidemiology on masks. You'll find it. And then all proceeds are going to be used to help COVID-19, and it'll be worth 80 points. And if you weren't here for the first class, basically what we are doing is a point system where the more you collaborate in Kid Teach Kid, or the more you're engaged in activities, then stop spamming, guys. I'm gonna mute the chat. All right. So the more that you, wait, is it muted? Okay, yeah, it's muted. Okay, so anyway, the more that you engage in activities in Kid Teach Kid, then the more points you get, and you can use these points to attend our special sessions. We have a lot of them. And the book that I published, if you buy it, it's gonna be worth 80 points, which is the most that we give out, basically. And also all of the proceeds are going to be donated to help fight COVID-19. So I'm not being greedy or anything. And also my book has been endorsed by a few professors and public health professionals. So it's a trustworthy resource. And I also highly suggest that you guys take the public health course. Where's the homework for public health? Oh, I'm sorry, Rich hasn't uploaded it yet, but it should be up by tonight. And also, if someone is asking me about points, basically, Rich will send out a more in detail document or email about points later today. So just wanted to give you guys a heads up about that. So yeah, you guys should all get my book, basically. It's not that expensive. I also recommend you guys get the ebook, since the ebook is, you can just download it and read it. Meanwhile, if you were to buy the paperback, it would take a while to ship and etc. So yeah. All right, so we're just gonna move on to the first thing we're gonna cover in class today, which is basic chemistry concepts. Here's the periodic table. I'm sure a lot of you guys are familiar with the periodic table. So we can see that over here, oh, sorry, my bad. There's the starting from boron, silicon, germanium, arsenic, etc. That line is called the zigzag line. Basically separates the metals from the non-metals, and everything that's on this zigzag line is a metalloid. So metals have shiny properties. They're shiny. They're for the most part malleable and ductile, and they conduct electricity very well. But on the other hand, we have non-metals like carbon, nitrogen, oxygen, etc. And these are not malleable. They're brittle. They're non-ductile, and they're not very luster. They don't have a lot of luster. So yeah. And whoever it keeps asking me to, no, okay. Anyway, so on the first group, the groups are going vertically, and then the periods are going horizontally. So, and one exception is hydrogen. Even though it's to the left of the zigzag line, it is a nonmetal. So group 1, which is the first row going, or the first column, those are all the alkali metals. They're highly reactive. And then the second is the alkali earth metals. And then the pink ones are transition metals. And then on the other side, where we have the yellows, those are the nonmetals. And then the light blue are the noble gases that don't react with anything. Okay, I'm gonna open the chat just for a bit, because I know like you guys like talking to each other, but please don't spam. Okay, that's basically all you really need to know about the periodic table. Yeah, we'll move on. So in biology, there are a few very important compounds and elements that are in the body. The most important one would be oxygen. As you can see in this body diagram, it constitutes 65% of the of a person's body mass, and not body mass, sorry, composition in their body. And then there's carbon, and then there's hydrogen, and then there's nitrogen, and then the other ones, they're very small amounts, but they include phosphorus, sulfur, etc. There's a lot of them, but they're not as important as the ones that are highlighted in large percentages. So atomic theory is shown on the right. Basically, for each atom, there are 3 components to an atom. There are electrons, which are on the outside. You can see they have negative charge. And then there are protons and neutrons in the nucleus. So protons have positive charge and neutrons have like neutral charge. They don't have a charge. Okay. Wait, sorry. My chat is kind of lagging. All right. So then basically what we have is the reason why the diagram on the left has like such a ball around the nucleus is because we can't exactly figure out where the electrons are going to be, because there's a principle called the Heisenberg uncertainty principle that tells us that we can't we have to compromise between what we know about where the electrons are. So that's why we usually just draw them as a cloud, since we don't exactly know exactly which energy level they're going to be in. And also, as part of this periodic table, we're going to look at boron, okay? Boron, we can see a 5 above it. That means the atomic number of boron is 5. So what that means is there's going to be 5 protons in boron and also 5 electrons. Yeah. And then neutrons, they can change, but once you have a different like number of neutrons for each element, then that means they're called isotopes. And to find the mass of an element, you basically add the number of neutrons to the number of protons. Okay, just to sum that up real quick, protons do not change at all. Electrons, you can gain them or lose them, but they are for the most part the same as the number of protons. And then the number of protons or the number of electrons is equal to the atomic number of the element. And the neutrons can change for each element. I mean, like they can be different for a single element, but when they're different, they're called isotopes. So for example, if we're going to look at carbon, we can see that carbon is 6. That means there's 6 protons, 6 electrons, and carbon also has 6 neutrons, but there are also different isotopes of carbon that have, for example, it can have 5 neutrons or 7 neutrons, etc. So that's when we have different like isotopes of the same element. And one more thing to discuss Also, please stop spamming, guys. If you're gonna spam, I'm just gonna mute the chat. All right, muting chat. All right, so anyway, what was I gonna say again? Oh yeah, so electrons, they're organized by energy levels, and if we look at this periodic table, the number of electrons on the very outside energy level is called the valence electrons, and that's the number of electrons that can actually interact with other elements to gain or lose electrons. And when an atom gains electrons, then it has a negative charge. Remember, electrons are negative, so when they gain, looking at hydrogen, or no, if we're looking at, let's say we're looking at fluoride, okay, fluorine, sorry. Fluorine, it can gain another electron so that it fills out its outer energy shell to have max capacity. And that means they gain, fluorine gains an electron, and that means it becomes negative. And whenever an element has a negative charge, then that means it is an anion. Meanwhile, if we look at sodium, we can see that it has one electron on the outer energy level. So whenever it loses an electron, it actually becomes more positive since it's getting rid of a negative. So that's why whenever sodium, it becomes an ion of sodium, and then it loses an electron, so it becomes a cation. Does anyone have any questions? That may be a little confusing, but if you guys have any questions, I will definitely explain it again. What's an isotope? Okay, so an isotope is the same element, but with different number of neutrons. So for example, if we're looking at, I'm not exactly sure if this isotope exists, but for example, carbon can have 6 neutrons or it can have 5 neutrons. Those would be examples of an isotope of carbon because they have varying number of neutrons, basically. All right, does anyone have any other questions at all? I'll answer them since I know that was kind of confusing if you haven't learned chemistry yet, basically. If no one has any questions, I will just continue. Oh, what are the numbers on the periodic table? Okay, that's a good question. So each number represents the element's atomic number. And what that means is that's the number of protons in the nucleus or the number of electrons that it has. So if we're going to look at carbon, the number is 6. That means that inside the nucleus, or you can see in this diagram, inside the nucleus, there are going to be 6 protons. And on the outside, there's going to be 6 electrons. So that is the number for each period on the periodic table. Can I list the, okay, are there always the same number of neutrons, electrons, and protons in an atom? The short answer is no, because neutrons can vary. Protons and electrons, for the most part, are the same. So let's talk about hydrogen. Hydrogen has 1 proton and 1 electron, but it has 0 neutrons. So protons and electrons are the same, but neutrons can change. That's why we have different isotopes. Hopefully that helps you answer your question. Can you explain the energy levels and how they relate to the electron shells, SP, D, and F? Okay, I'm glad that you know about the SP, D, and F, but we're not going to be discussing that in this class since I just wanted to cover the fundamentals of chemistry. I think Etienne may cover it in her class. So if you're interested in learning about that, attend the chemistry class, okay? All right, next we have polyatomic ions. Polyatomic ions, I think I mentioned earlier that ions are basically when an atom gains or loses an electron so that they have a positive or a negative charge that's not neutral. So polyatomic, it means there's more than one atom. So on the right, we have a list of many polyatomic ions. You can see for each of them, there's more than one atom and they all have a charge. A lot of them are negative, but ammonium, the second to last one, is positive. So these are just some common polyatomic ions that are going to show up in biochemistry. If anyone has a question about a certain one or wants me to go over a certain one, I will definitely talk about it. All right, next we have chemical bonding. This is where we actually start getting into biochemistry. So the first type of chemical bonding that we will talk about is ionic. So what ionic means is you can just look at this diagram right here. You can see that sodium has 1 electron on its outer shell, meaning that it has 1 valence electron. But now let's look at chlorine. Chlorine has a -1. It's missing 1 to complete its outer shell. So if sodium donates its outer electron to chlorine, then sodium can have a full outer shell and chlorine can also have a full outer shell. So this way there's like an exchange of electrons. That's how we know that it's ionic. So here are some common examples. Sodium chloride, which is salt. That's the thing that we're showing on the right here. And then there's sodium hydroxide. That may be kind of weird for you to understand, but let's take a look at sodium hydroxide. If you see the OH, that means hydroxide, and in here, hydroxide is not written in the polyatomic ions, but basically hydroxide itself is the OH has a negative charge, so it's missing one electron. There's 1 extra electron on its outer shell, so then it's missing 1 electron, so sodium can donate its 1 electron to hydroxide. So that's why it's ionic, even though it's more than 1 compound. Is there any specific way to abbreviate the elements on the periodic table? Yes. So as you can see here, these are all the element abbreviations. So instead of writing out hydrogen, you can just write H. Instead of writing out chromium, you can just write Cr. Yeah. All right, let's move on to the next. Oh, yeah, and same thing for calcium, CaCO3. Calcium carbonate. Basically, carbonate CO3 is a polyatomic ion with a negative charge, and calcium has a positive charge. So that's why it's ionic, because there's an exchange of electrons, 2 to be exact. All right, so next we have covalent bonding. And in contrast to ionic bonding, covalent bonding is when there's a sharing of electrons. So let's look at the examples on the right. So on the very top, we can see that hydrogen has 1 electron, another hydrogen has another electron, but they want to complete the outer shell, which needs 2 electrons, so they'll just share the 2 electrons with each other so that they can both complete their outer shells. And that's when they have a single bond, because each one is contributing 1 electron. So the first hydrogen's contributing 1, the second one's contributing 1, that's why there's a single bond. Okay. So I'm gonna open the chat again since it's been a while. But if you guys have any questions, please feel free. Oh wait, my computer is lagging. Or not. Okay, anyway, let's move on to the oxygen activity, the oxygen example. We know that oxygen has 2 it's missing 2 electrons on its outside. So that's why you can see that they're sharing 2 electrons with each other. The first oxygen is giving the second one 2 of its electrons, and the second one is giving the first one 2 of its electrons so that they can share amongst each other to complete the entire outer thing. Wait, chat is still disabled? Yeah, I know, I'm sorry, because my computer is lagging. Someone's asking me, how does this relate to biology? I think I mentioned earlier, we're covering biochemistry today. And in order to be able to cover that, we need to set a good understanding for chemistry first, just in case some of you guys may not know these fundamental chemistry concepts. Okay, I'm gonna turn on the chat right now. Okay, it should be turned on. All right, and then similarly, there's water, and then there's carbon there's methane. Those are all examples of covalent bonds. Someone asked me, can I go over valence electrons again? Yeah, sure. So basically, for each element, there's going to be various energy shells. On the very outside, there's going to be, the very outside energy shell is the shell that the electrons are actually interact with each other, gain electrons, lose electrons. So basically the electrons on the very outer energy shell are going to be the valence electrons. Hopefully that makes more sense for you. Okay, we're gonna move on to hydrogen bonds. Hydrogen bond is not a real like type of like a real compound bond, but basically here's where I'll introduce the concept of electronegativity. So electronegativity is the ability for an element to attract electrons. So in this case, we have a water molecule on the right. The water molecule has an oxygen in the middle, and oxygen has a high electronegativity, meaning that it has a high potential to attract electrons. So oxygen attracts the electrons in hydrogen more than hydrogen attracts its own electrons, which means that hydrogen will have a net positive charge. The reason behind this is because electrons are negative, so if less electrons are going to be near the hydrogen, then the hydrogen will have a positive charge, if that makes sense. Also, please stop spamming in the chat. It's gonna be hard for me to see any like concerns that anyone has. So yeah. All right, next so anyway, about hydrogen bonds. Since the oxygen will have a net negative charge since it's more likely to pull electrons, and the hydrogen has a net positive charge since it's less likely to pull electrons, then we can see that the same case happens with the ammonia ion. So the net positive charge in hydrogen attracts the net negative charge on nitrogen, which forms a hydrogen bond. That's basically how hydrogen bonds work. Does anyone have any questions about that? Can an alkali earth metal react with a halogen, and if so, what happens to the excess electron? Okay, so that's a good question. Basically, you need two halogens basically. So if we're going to go back to the periodic table, if we have magnesium. Okay, so for example, an alkali earth metal, let's take magnesium, okay? And then let's take a halogen, let's take chloride. So magnesium can react with chloride, but the compound will be magnesium. So one magnesium and two chlorides. This way, the magnesium can give one of its valence electrons to one of the chlorides and give the other one to the other chloride. So that's basically how an alkali earth metal reacts with a halogen. Can hydrogen bonds only be polar covalent? Hold on. Well, in here, hydrogen bonds are non-polar covalent. And what that means is, they can be polar covalent or they can be non-polar covalent. And if you don't worry about that, I'm not going to worry too much. I just wanted you guys to know what a hydrogen bond is, okay? So if anyone has other questions about hydrogen bonds, please let me know. And please stop spamming the chat unless you're trying to help me promote my book. But yeah. Okay, anyway, next we're going to go on to other intermolecular forces. And hydrogen bonds are examples of intermolecular forces since it's between one molecule and another molecule. So van der Waals would be an example of an intermolecular force. So I think I told you guys earlier, in a compound or in a element, the electrons, we're not sure exactly where they are. So they could be in one area all of a sudden, or they can be in another area all of a sudden. So if they're in one area all of a sudden, then that area has more of a negative charge. But if another element and they're in another area, then the negative charge of one area will attract the positive charge of another area. This is just based on chance, basically. So yeah. These are very weak because they're not often found. So there are better ways to have more strong intermolecular forces. For example, electrostatic interactions. Basically, just wherever there's a negatively charged element and a positively charged element, or partially positive, they attract each other. And then there's hydrophobic and hydrophilic. Hydrophobic means it does not like water, and hydrophilic means that it does like water. So for the most part, the hydrophobic areas will clump together, and then the hydrophilic areas will go on the outside. So yeah, that is an overview of all the intermolecular forces. Does anyone have any questions? Okay, can you guys like please not, okay, it's okay to talk in the chat, but just try not to like spam too much, okay? All right, next we have organic chemistry. This is where there's going to be more memorizing. So organic chemistry is basically chemistry relating to carbons. And carbon, since everything living requires carbon, we term the chemistry of carbons organic chemistry. So we can have two carbons linked to 1, 2, 3, 4, 5 hydrogens, or sorry, 6 hydrogens, my bad. So there's various, and based on how long the carbon chain is, there is a different name. So for example, if we have 2 carbons, we call it ethane. If there's 3 carbons, we call it propane, 4 butane, etc. And then on the, here we have, if we have a double bond, then that means that the carbons are sharing electrons, so they're not sharing with hydrogen anymore. So that means we don't need as many hydrogens. And do you guys remember what hydrogen bonds are? If you guys do, do you guys remember what double bonds are? If you guys don't, I will go over them again. Stop spamming. You guys remember what double bonds are, right? Okay. If you remember, then I'll just keep going. That's basically when each element gives the other one 2, and then the other one gives the other 2, and they share it, basically. Instead of a single bond where they share, each one gives the other one 1. So yeah. All right, next we have functional groups. Don't worry too much about functional groups. These are more for advanced organic chemistry, but I just wanted you guys to know. So for example, if you see these little bends in the functional group, so for example, for alkane, you see there's a bend kind of like a V on the outside. The bend means a carbon. And basically, yeah, that's really it. Each bend represents a carbon, but since there's going to be too many carbons to write, that's why there's we use bends to represent them in organic chemistry. And R, you may see a lot of R's in here, and R just means the side chain, just means another group on the outside. It's like basically anything you want, basically. R just means the side chain. All right, so next we're gonna move on to the fun stuff, which is actually biochemistry. I know you guys have been looking forward to this part for the entire class. So macromolecules. Macromolecules, the definition would just be like molecules of carbohydrates, nucleic acids, protein, or lipids. Okay, so we're gonna talk about carbohydrates first. So carbohydrates, the monomer of a carbohydrate would be a monosaccharide. So what a monomer means is, let's break it down like this. Macromolecules, it means a lot of molecules strung together. So each one of those small things that are strung together to create a macromolecule, each one of those is a monomer. So for example, for carbohydrates, we have lots of small monosaccharides being strung together to create a sugar, which is a carbohydrate. Does anyone have a question about that, or wants me to re-explain any part of what I just said? Okay, I'm gonna pause the chat so you can't start the spam contest. Okay. All right, so anyway, we have monomers of carbohydrates would be the monosaccharide, and then a dimer, meaning 2 monosaccharides combined together, 2 monomers combined together, that would be a disaccharide. And then multiple monomers combined together would be a polymer. So that would be, in the case of sugars, it would be a polysaccharide. Does that make sense to you guys? Yeah, sure, I'll definitely repeat it. So the reason why we call the reason why it's called macromolecules, it's because they're large molecules. Macro means large. And to make large molecules, we need to string together a lot of small molecules. So each one of these small molecules that we string together is called a monomer. So in the case of carbohydrates, the small molecule would be called a monosaccharide. So many different monosaccharides strung together would be called a polysaccharide, and that would also be called a sugar, basically. Okay, So used as a temporary fuel source for the body. And monosaccharides are in the form CnH2 and ON, and where C represents carbon, H represents hydrogen, and O represents oxygen. So on the right, I have an example of glucose, which is a monosaccharide. Glucose is just one small monomer for sugars that it makes up. So glucose, the formula is C6H12O6, and when you string two glucoses together, we make a dimer, which in this case would be called sucrose. And you may be wondering, oh, why is sucrose not C12H24O12, since we're stringing two of them together? You'll see this in a bit where I explain how to create bonds with different monomers. What does the N mean? That's a good question. So N is basically the number of carbons in a monosaccharide. So for example, in glucose, there are 6 carbons. So that means the form will be C6H12O6. So yeah, hopefully that helps you a little bit more. Is it normal that you know this? Good for you if you know this. That's great. All right. So now we'll talk about some monosaccharides. So over here we have glyceraldehyde. It has three carbons. So we can see the formula would be C3H6O3. And then we also have 5 carbon sugars where the formula would be C5H10O5. So yeah. And notice that there's always going to be a double bond between carbon and oxygen. So, but it can be in various places. So yeah. And then now let's talk about how we can form rings with monosaccharides. But does anyone have any questions as of now? No? Okay, cool. So you can see that over here, we take the long string of carbons and then we can combine the pink area with the blue. Okay, you know what? It's kind of hard for me to explain this. So go ahead and look at what's going on right here. And if I can answer any questions, I will do that. But basically, yeah. Okay. Okay, the double lines means a double bond. So a double bond is a type of covalent bond. Let me go back up. Where was it? I think it was here. Okay, so the double bond is so you can see it over here in the oxygen part. The first oxygen is giving the second one 2 electrons, and the second one is also giving the first one 2 electrons, and they're sharing a total of 4 electrons. That's why it's a double bond. But contrast that with the first one, where the first hydrogen is giving the second one 1, and the second one is giving the first one 1. There's a total of 2 electrons being shared. That's why it's a single bond. So yeah. Don't worry too much about the details about this. I just wanted to let you guys know, be sure to know about what carbohydrates are and what monosaccharides are. So if you guys have any questions, feel free to ask me in the chat. When did this meeting start? It started at 5 o'clock. Okay. So next I'll show you guys how we can link monosaccharides together to create disaccharides or polysaccharides, etc. So if you see over here in the 2 glucose molecules, the first one, if you flip it over, you'll get the second one, and we all know that water is H2O. So you take the OH from the first one and the H from the second one, and then those two combine together to create a water molecule. And then in the meantime, there's a bond that's created that uses the oxygen to link the two monosaccharides together and to form a disaccharide, basically. Oh, sure. I'll go back to the trioses and stuff. Here, go. Is there anything that I can answer for you about the here? Since you guys can type it in the chat, but just don't spam. All right. So now I'm going to go on to the next type of macromolecule, which is lipids. So earlier you may have seen in this that that carbohydrates only have 3 elements: carbon, hydrogen, and oxygen. This is the same for lipids. Lipids also only have 3: carbon, hydrogen, and oxygen. So, but they have structural differences. So we have 2 different types of lipids: unsaturated and saturated. Saturated means that there's one double bond inside. So if you look at this first diagram over here, we can see that there's a double bond, and that causes the lipid to be unsaturated. But on the right, you can see that there are no double bonds. Everything is single. Everything is a single bond. So that's why it's a saturated lipid. And all lipids are hydrophobic, meaning that they hate water. They don't mix well with water. And you guys probably know that, since you know that you can't mix oil and water. And lipids function for long-term energy storage. Okay, actually, that's actually a great idea, Stephen. Let me change it to all panelists. Sorry, my computer is kind of lagging, but I'll change it whenever. Okay. There we go. I think that works now. Okay, so the function for lipids would be to store long-term energy. Oh wait, oops, I did not do that. But anyway, unlike carbohydrates, where it only functions for short-term energy. So short-term energy, for example, if you just ate like bread and you want to go out for a run, your body would use carbohydrates to give you that energy. But on the other hand, if you run out of carbohydrates, then that's when your body starts going into the lipid supply and begins to use that. And your liver is a good source of storing fats for whenever you need to use the long-term energy supply of lipids. So yeah, and also here I have a list or a chart comparing saturated with unsaturated. So an example of saturated fat would be like animal fat or butter, where it's in room temperature, it's solid. And the reason for that is because since they're all single bonds, they're super compact together. There's no like kinks. See, look, for example, for here, you can see that the unsaturated one goes down, so they're not like a straight line. That's why they're not as compact. But saturated, you can see that they're all straight lines. So when you pack them together, they're very tight, and that's why they become solid, like butter. But on the other hand, if we have unsaturated, there's at least one double bond in there. So for example, olive oil, it's a liquid because it's not as compact, because they like tend to kink and do like weird angles. So they're not as compact, and that's why they're liquid. So here we have solid at room temperature, and for the most part, saturated are very unhealthy, and unsaturated are more healthy since it's easier for your body to digest, basically. Does anyone have any questions about what I just went over regarding saturated and unsaturated fats? Oh, trans fats. That's a good question, Max. So basically, you can see how the lipids in the unsaturated fat here are bending. That's because they're in the cis conformation, meaning they're a cis unsaturated fat. But sometimes, like commercially, they can engineer fats to stay straight and not kink around. So they become in the trans configuration, and that's why they're called trans fats. And trans fats are extremely unhealthy. They're even more unhealthy than saturated fats, which is why most foods try not to use trans fats. It mostly depends on the conformation that the carbons are in, whether or not they allow them to bend. So yeah, that's a great question. All right, so now we're going to move on to my favorite macromolecule of them all, proteins. So in proteins, we add another element, which is nitrogen. So if you remember earlier, we have carbon, hydrogen, and oxygen for carbohydrates and lipids, but now we're adding nitrogen because proteins can use that extra element. So on the right, we have the basic diagram of a protein. If you go on the left, if you see this, the black diagram, that's all black. you can see that on the left, there is an N and then two H's, and that's called the amine group. And then on the right, you can see there's C with the double bond to O, and then there's, it's bonded to a hydroxyl group, and this would be called a carboxyl group. So every single amino acid, which is the monomer of a protein, has this structure, okay? And then the R means the side chain, so that part varies between amino acids. Sorry, I didn't really go over amino acids, but that's basically the monomer of proteins. Raymond is asking, is there going to be another class after this one, or is this the last one? For today, this is the last one. Okay, so now we're going to talk about how we can combine multiple amino acids to actually make proteins. So this is very similar to carbohydrates. You can see the OH and the H, and then they combine together to make water, and the water comes out, and then they form a bond with each other called a peptide bond. And when you have multiple peptide bonds, then that when you have multiple amino acids joined by peptide bonds, you have long chains of amino acids called polypeptides, and even longer chains called proteins. So that's basically how proteins are formed. Okay, does anyone have any questions about proteins? Okay, here are some functions of proteins. Proteins are some of the most versatile functions, which is why they're my favorite macromolecule. So for example, an enzyme, proteins make up most enzymes in your body. So one of the enzymes that I like is called disulfide isomerase. So basically, disulfide isomerase is a enzyme that helps proteins fold, which I think is really cool. And then they can serve as storage. Basically, casein is a type of protein that stores nutrients in embryos of non-humans, basically. And then hormones like insulin. Insulin is a protein. It helps keep body sugar low. And then there are structural proteins that basically make the structure of your body. So for example, your skin is made of keratin, your hair is made of keratin, your nails are made of keratin. They're basically there to serve structure for your body, and keratin is a protein. They can also be receptors, like for example, in neuromuscular like in neurons, between neurons, receptors that receive the neurotransmitters, those receptors are proteins. And they can also serve as to transport nutrients. And what that means, for example, we can use the example of hemoglobin. So you guys may have heard of hemoglobin. Hemoglobin is a protein in your blood that is used to transport oxygen. So yeah, basically it transports oxygen. Otherwise, oxygen will not be able to be in your blood since oxygen is not soluble in your blood. But that's what in your plasma. So that's why we have hemoglobin, to allow oxygen to bond to it and transport the oxygen. So that is how proteins can serve as transport. And also, I'm not sure if you guys knew this, but antibodies are also examples of proteins, and they serve to defend the body against attacks of foreign pathogens and other not a good things, basically, for your body. So yeah, if anyone has any questions about the various functions of proteins, just feel free to let me know. Okay, so since I don't see any questions right now, I'm going to move on to the final macromolecule, which are nucleic acids. And I'm just going to go over this one real quick. So nucleic acids are DNA and RNA, and the monomers are a nucleotide. And in each nucleotide, we have different bases. So we have adenine, thymine, cytosine, and guanine. And those adenine combines with thymine in DNA, cytosine combines with guanine in DNA as well. And purines that are adenine and guanine, that just means it has two rings basically in its structure. And then pyrimidines are cytosine and thymine. They have one ring, and they're combined to each other by hydrogen bonds, as you can see in this diagram on the right. So yeah, here we have a quick little comparison between DNA and RNA. So you guys can go ahead and look at that. Basically, DNA is double-stranded, RNA is single-stranded. And in RNA, instead of having thymine, there is a new base called uracil. They're very similar, except uracil, I think it just has like one less methyl group or something like that. They're very similar. But in RNA, it has uracil instead of thymine. Okay, so now we have that RNA, there's many different types. For example, you may have heard of mRNA, which is messenger RNA, that's used for transcription. That's transcribed to mRNA and then used for translation of proteins. And then there's tRNA for transfer RNA, rRNA for ribosomal RNA, etc. There's a lot of different types of RNA. So yeah, now we're going to move on to molecular biology. We only have 10 minutes, but we will be able to cover most of for this, all we're just going to be talking about is the various organelles in a cell. So first we're going to start from the top. So we have the endoplasmic reticulum that basically surrounds the nucleus in an animal cell, and it manufactures ribosomes. It helps with detoxification. It does a lot of stuff. And the rough ER has a lot of ribosomes on it. The smooth ER does not. Okay, and then we have the nucleus, which is probably the most important part of the cell. And keep in mind that only animal cells and plant cells my bad. Basically, all eukaryotic cells have nuclei. So that includes protists, animals, plants, fungi, yeah. But then there's bacteria that are non-nucleated, and archaebacteria, they do not have a nucleus, so they're prokaryotic. And then on the inside of the nucleus, there is the nucleolus, which is used to manufacture ribosomes. And ribosomes are used for creating proteins, basically. They take the DNA information and create proteins. Next, we have the Golgi apparatus, which is used to transport various nutrients inside the cell. It is also used to synthesize some nutrients and etc. And then we have the lysosome, which is used to detoxify various toxins inside the cell or various waste materials. It has a lot of strong acids that are used to neutralize these various toxins. And then we have mitochondria. I'm not going to say the cringy powerhouse of the cell thing because it's weird, but basically it generates all the ATP for the cell. It creates all the energy needed for the cell to perform its functions. And then we have things like the plasma membrane. It basically covers the entire cell. It's made up of, here, let me show you the structure of a plasma membrane. It's this blue thing right here. There's, on the outside are hydrophilic heads, so water can touch the heads, but water does not like the inside of the plasma membrane, which are the hydrophobic tails. And then we have the flagellum, which is used, this is not in all cells. Not all cells have a flagellum. It's used for motility, which means it allows the cell to move. Is there anything I'm missing? There's things like cytoskeleton, which keeps the structure of the cell. Oh, cytoplasm. Cytoplasm is like the jelly-like fluid inside the cell to make sure all the organelles are in their place, yet they can still move at the same time. And then there are things like microfilaments that are also used to help the cell move and keep its structure. And then there's the peroxisome used to create hydrogen peroxide as a byproduct of various metabolism activities, metabolic activities. So yeah, if you'll have any questions about what I just talked about. Oh, someone asked, isn't that mobility? The thing is, motility and mobility are almost interchangeable in talking about cells. So yeah, you can basically use either one, but there are some minor differences. I can't remember them off the top of my head, but for the most part, for cells, we use motility. Max asks, why do skeletal muscle cells have more than one nucleus? That is a great question. So think about it. Your skeletal muscle cells allow you to move, and they generate a lot of, they have a lot of first I'll talk about why they have a lot of mitochondria. That's because they need a lot of energy to move. And that, I think that's the reason for the mitochondria, but for the nucleus, it's just like a structural adaptation for, I think it's just a structural adaptation, but there's not really like a huge, okay, correct me if I'm wrong, but I don't think there's like a huge reason as to why there's multiple nuclei It's just that the nuclei are on the outside of the skeletal muscle so that the inside can be made of sarcomeres so that they can contract and move. I don't know if that answers your question, but it's like mostly as just a structural adaptation. But as for mitochondria, there's a lot in order to generate a lot of energy. Okay, so next we have plant cells. There's really only a few major differences. It retains most of the same organelles as animal cells, except now there's a large central vacuole to make sure that the plant cell is fully like stretched basically, so that there's turgor pressure. You can see that it's like a huge clear thing in the middle. And then there are also things called chloroplasts that contain chlorophyll. And this is used for photosynthesis for the plant to generate sugar from light. And then aside from the plasma membrane, the cell also has a cell wall that is used to stabilize its structure, basically. Yeah. There are also these holes in between cells walls called plasmodesmata that allow like nutrients, sometimes liquids, to flow through and allow cell to cell like transfer of nutrients. Okay, so that's about it for our class today. If you guys have any questions at all about any of the things that I talked about, please ask me. I will answer them, hopefully. But also before you leave, I just wanted to recommend my book again for those of you guys who came in later. I recently published a book. I have information about it here. It's on epidemiology and public health. And it's a very good resource for you to help if you're dedicated to it. And also it's basically it's the textbook for our public health class. I'd say the public health class is a really good way for you to learn about COVID-19 and how to stay safe in the population and how diseases transmit through populations. So yeah, and then you can visit that on the website. Oh, so okay, so the ebook is $4. I would recommend you get the ebook because it's easier for you to just see. But the actual book itself, like the paperback book, is $9, but I would recommend the ebook itself. So yeah. And here, yeah, it's a pretty good book. I'd say I spent a lot of time on it. So if you guys would help me out and buy it, I would be very thankful. And all of the proceeds are going to be donated to help fight COVID-19. And also, it's worth 80 points. And if you don't understand what points are, don't worry. We're just gonna send out a email talking all about points and how you can earn them and how you can use them for the future. So yeah. I hope you guys really enjoyed this class and learned something new from it. And if you have any questions, I'm still here. You can ask me. So, yeah. Oh, by the way, I'm going to open the chat right now so you guys can talk if you want to. I think it's open. Yeah, it's open. The point system is not pay to win. This is the only actual activity that requires money to earn points. The rest is just like going to a class or attending homework, etc. Yeah. And also, if you guys like the class today or my public health class, feel free to recommend Kid Teach Kid to any of your friends or my book to any of your friends. That is also another point earning move. So I would really, really appreciate that if you guys could help us out. So yeah. And if you have any other questions, you can just ask me in the Q&A, I'll answer them. But if not, you guys can leave. Thank you for a great class. Thank you for being respectful for those of you guys who do not spam. Oh, one more thing. We do have homework, but it's optional, but it earns you points if you do them. So it's not up yet. Rich will add it to the Kid Teach Kid website. It's not that hard. It's pretty easy. So it's optional to help you understand like exactly what went on in this class today. So yeah. Yeah, please buy my book, guys. It's a very great book, I think. And it would really help me a lot. Oh, okay, don't worry. So after you buy your book, you can fill out a form with all it'll ask you to do is fill out the order number, and then I'll just go back and check to see if your order number matches up with a real order number, and then you'll earn points. that's basically how we, oh, the website is just going to be kidteachkid.org. To buy the book, just go on lulu.com and search epidemiology unmasked. You should be able to find it. And the points, yes, class is over. The points, Rich is going to send out a more detailed information email about it, so that'll be that, and you'll see more about it. But basically, it'll let you attend special sessions hosted by like some really OP people, so yeah. Homework, it'll be posted later today on the website, and then submitting it, I think you can just, there'll be more informations. You can just, I think you can just email us. I'm not, but Rich will talk about it in his email, so yeah. So thank you guys so much for coming. If there are no more questions, I'm gonna go ahead and end this chat. It was great, end this meeting. It was great getting to know you guys. Bye.